Hi everyone, Mrs. Ortega here. I am getting ready to start our newest material. I'm very excited as our next material in Exam 4 and then later in Exam 5, we're going to transition from evolution of plants to animals. So that's what the next material will be all about. Will be animals, animal diversity, forms and structure. And then in Exam 5 material, we'll be looking at human systems. So these chapters, these last two exams are usually a little bit better for students as they're more familiar with the topics already before we get into them. And so right now we'll start with chapter 32, which is an overview of animal diversity. In this chapter, we will look at some common unifying themes of animals, along with some developmental structures. All right, first starting out, you'll want to think about how do you define animal? What would make up an animal? What would not be an animal? While it seems pretty easy, it's not all that easy, as some animals have fur, some have scales, some have 4 legs, some have 2 legs, just a very, very diverse amount of features, along with possibilities in terms of nutrition, food, and in terms of abilities like speed, strength, camouflage. So that's where it starts off here. That most animals are mobile and they use different traits to detect, capture, and eat other organisms. The chameleon in this image here would use its long, sticky, quick-moving tongue to capture its insects, and use this great ability of camouflage to go undetected by larger predators. Right, so overall, though, animals, we define them as being multicellular, heterotrophic eukaryotes. So all animals will be multicellular. All will ingest food from outside their environment, so that would be heterotrophic, and then eukaryotes contain eukaryotic cells that have membrane-bound organelles. And so they will also have tissues that will develop from embryonic layers, and that's another common theme we'll see. So their nutritional mode are heterotrophic, and they ingest food from outside their environment. In terms of cell structure and specialization, they are multicellular eukaryotic cells. They do not have cell walls, which is a unifying trait of all animals. Their bodies are going to be held together by structural proteins and structural tissues like collagen. They also have unique tissues such as nervous tissue and muscle tissue, are different types of cells working together for different functions, and so they will act as what we call a functional unit. And so those are also unique to animals. Okay, so we're going to look at reproduction development. So that embryo, once it's formed, how does it undergo some development? And so it starts out with the gamete cells, the sperm and the egg. Once they fertilize, once they fuse together, the zygote is formed. This will undergo rapid cell division, and that rapid cell division is called cleavage. And so that cleavage will lead to a formation of a large hollow ball of cells called the blastula. And so after that, then it forms a secondary layer of cells, and that will be a process called gastrulation with the formation of the gastrula. And so I believe this next slide here has a nice image. And so here you can see the zygote is an egg and the sperm cell fused together, and now forming is now a zygote is going to form embryo. And so undergoing rapid cell division or state of cleavage, it'll have an 8-cell stage there of zygote cells. and then undergoing cleavage more, then you form a larger version, essentially, where there's a hollow cavity, and that's inside the ball of cells, which is the blastula. And so here the blastula is something should be on your review. And you can think blastula starts with the letter B. Also, what starts with the letter B is ball. So hollow ball of cells. So that cavity that I was talking about that's inside the blastula, that's called the blastocele. And so this word, while not super important, we will see in the embryonic development different words for cavities, which is something that's important in this chapter. And so this word, while not essential to this part, is something I do like to point out. The blasto coming from the blastula in terms of that first part, blastula, seal, in this form means cavity or opening. Not like the teeth cavity. Cavity is just a opening region where there's really nothing there. So seal means cavity, and you'll see that in this chapter. So after the formation of the blastula, the hollow ball cells, then we go to the gastrula. And you can think alphabetically, B is before G, so blastulation happens before gastrulation. And so that second layer is being formed by pinching inward of a region. And so once that happens, then now you have 2 layers of cells. And here you can see it's labeled. The outer cavity is the blastocele still, and then the inner cavity is now called the archenteron. And so that's the inner cavity with that yellow region. And so the layers, the cell layers, are now given names. The outer one is ectoderm, like we saw before. Ecto is meaning outer, and so outer, and then derm, like dermis or skin. So this is outer skin, and then endo is inner, and so that means inner skin. And so if you know the breakdown of the words, you really don't have to memorize them, which is what I try to teach you. So this is something you'll want to understand for sure. As I go through, I'm still going to try to make sure I correlate everything tightly with the review, which I have with me. Other things that we saw earlier was defining animals. That's something you'll want to know, so I'll go ahead and show you and mark that page. So this is a good page to refer to in terms of definitions. Let's see here. This concept is something you want to know. All right. Didn't get that far. All right. Moving along to in development. So after that gastrulation, then there's more complexity in terms of growing from that egg, and eventually will reach what we call a larval stage, or a juvenile stage. So there is a difference for us, larva and juvenile here. Let's start with the larva stage. This is for animals that will have a morphologically different body plan as they're growing, as they're young. And so they have to undergo a physical change called metamorphosis to become a juvenile. And so it's kind of a precursor. So you have the egg that's growing, and then you have the larva stage where that organism is distinctly physically different than the adult version. And so after that larva undergoes a metamorphosis, a physical change, then it'll become a juvenile, which is not pictured on this lovely graph or chart here. Juvenile, which is going to have the same physical appearance as the adult, but it's not yet sexually mature, so it has to undergo development in the sexual reproduction department before it becomes the adult. And so that's something you want to understand. Larva is distinctly morphologically different. So there's two things that separate it from becoming an adult, is its physical appearance. Just kind of abbreviate here, physical appearance, and then it's the sexual reproduction capability. And so that's the second thing. And so for juveniles, they have just one of those to overcome. So that is something you'll want to remember. All right, moving right along here. Other things in terms of genes and genetics, the Hox genes regulate body form. So this is, most animals will have these Hox genes. Only animals will have Hox genes. And so that, from a genetic standpoint, is something that does help classify animals. In terms of history, animals have a very long history of more than 500 million years. So there are more than 1.3 million animal species that have been named, but far more are in existence, not discovered or named yet. And they believe the common ancestor of all living animals to be about 700 to 77 million of years ago. So remembering that origin of life on Earth section, this is before plants moved to land. Animals likely lived before then. And so looking at the origin of the multicellular animals, we kind of go back to our protist group, and in the 4 supergroups, one of them is the Uniconta, if you remember that word. That was a group that animals was coming from that tree, and so from Uniconta also comes a protist, and that is shown here, the choanoflagellate. So that's the closest living relative to animals that's not part of the animal group. They are part of the protist group, and you can see their cell closely resembles a color cell in the sponges, and so that's something that we'll look at in terms of sponges when we get to that section in the next chapter. How the sponge's collar cell closely imitates that of the choanoflagellate. And so in terms of origin, the choanoflagellate protist is very important for animals. And so studying that here, you would see that there are new ways for the cells to attach and signal or communicate with each other. And so those processes come from proteins. So there are proteins that help the cells adhere to one another as well as communicate. And so those are actually called cadherins. I hopefully have this spelled correctly. Cadherin protein is what helps anchor it. And so looking in terms of that genetically, you can see in this image below the lines that the choanoflagellate, and it's not very well labeled in your book, but I believe it should be what they illustrate as these here, have cadherin proteins that help anchor the cells to each other, also found in the animals. And so these are all very similar, if not the same, whereas animals have this what we call CCD domain. It's a very specialized cadherin domain that allows for a unique attachment structure. And so it doesn't have that in the choanoflagellate. As you can see here, they've color-coded it a different color in that region. I'm not exactly sure what they have in place of that CCD domain, but it is something different. So In terms of genetically speaking, they do have some shared region, but animals have a unique CCD domain that separates it from the choanoflagellate. In terms of eras, looking at eras is not something I'm a huge fan of, but if you're interested, there are lots of fossil evidence regarding animals, and there's a lot of information that is very interesting. This is not something I like to try to test you guys on, but we'll briefly go through it here. That the Neoproterozoic era from 1 billion to 542 million years ago had what we call sorry, let me think, lost my train of thought here. They had some animal fossils and evidence of predators in that environment. And so here you can see below in this fossil, this shelled, what believed to be a shelled animal has a borehole in the center of it that suggests that maybe an animal attacked it or a different predator attacked it at some point. But in terms of fossils from what we call the Ediacaran biota or era, which is here, they're mostly known for their soft-bodied animal fossils that resemble mollusks or snails or shells, and so that's shown here. So these, you can tell, are not super detailed or fine, but you can do make out a body plan. And so that illustrates that most likely these are soft body animals with some hard parts that are able to fossilize okay. And you will want to compare that to the time of the Cambrian explosion, which was around the time of the Paleozoic era, that had a major groups of living animals come out of this era. And most of them have the bilaterian structure, which is a group of animals that have what we call a bilateral symmetric form. That's something we'll look at. A complete digestive tract, from mouth to stomach, intestines, anus, and then it's a one-way digestive system, so it does not enter through the mouth and leave through the mouth. It will go one way. And so here in this next image, you can see a Cambrian seascape, like an artist's rendition of what it may look like based on fossil evidence and other records at that time, and then a fossil from that time period. And so in the Cambrian explosion, these animals had hard body parts like shells and spikes, and so that kind of marks a difference between the Cambrian explosion era and then the Ediacaran era. Soft body animals versus hard body animals. Again, this is all very important information. It's just not something I'd like to dig into in regards to testing and such. Let's see, in terms of timeline, they had in that explosion, the Cambrian explosion, new predator-prey relationships formed. There was also a rise in oxygen in the atmosphere as plants have moved onto land. And then at this time, I believe the Hox gene complex started to evolve. And so around 450 million years ago, animals made impact on land, and then vertebrates made the transition to land about 365 million years ago. And so looking in the Mesozoic era, that middle era, there are lots of coral reefs that emerged. There were reptiles that returned to water. Dinosaurs were dominant during this time on the land, but mammals emerged, flowering plants and insects diversified as well. And then the most current era, the Cenozoic era, had the extinction of terrestrial and marine animals. Most notably, the dinosaurs became extinct at the beginning of this era. 65.5 million years ago. And so as they left, that allowed for the mammals that were left behind to exploit those vacant ecological niches or ecosystems. And so they diversified. All right, so moving right along to the next key idea in this chapter will be how animals are characterized into body plans. So a body plan is defined as a set of morphological and developmental traits. And some of these are conserved throughout time, others change multiple times over the course of evolution. And so we will first define them, and then as we go through next couple chapters, we'll look at which body plans they have and how that, if that changes in terms of development for that organism or if it changed over time. And so body plans may or may not have symmetry. For sponges, our first animal we'll see, they do not have any body symmetry, one of the few. But most either have a radial or bilateral symmetry. So radial, like the radius of a circle, if you cut it in half, no matter which way you cut it in half, as long as there's equal amount on each side, it's going to be equal. And so that is what you see here in this flower pot example, sea anemone example. Anything that has a circular nature, and when you cut in half, it's going to be on the same on each side. That's going to be radial symmetry. And then the bilateral symmetry can only be cut in half in one way for it to be equal on both sides. So here we have a shovel and a lobster as our examples. Humans, we have bilateral symmetry. We can only be cut down the middle from head to toe to have symmetry, left and right arms, left and right legs. So that's something you want to understand. As we'll see, most animals have bilateral, but some will have radial symmetry. And for the bilateral symmetry, you have more definitions to go with that to help you clarify when you're discussing different aspects of these animals. That there is a dorsal or top side and a ventral or bottom side. In a region of, for example, like a dolphin, the dorsal top side is going to be the darker top part of their back, their head, their tail, and then the ventral or the bottom side would be like their belly, their underside. That's white. And so that's what I like to think of. Usually dorsal for animals will be a darker surface, and ventral will be like a lighter, whiter surface. But that's not always the case, so just keep that as a general trend. Left and right side, like left and right arm, left and right fin, left and right hoof. That's pretty self-explanatory. And then the anterior and posterior, so front end and back end. And so this is easy to think about us in terms of our anterior. That's our, for our head region, that would be our face. For posterior, that would be the back of your head. So we think for our dolphin example, their anterior would be their head region, their posterior would be their tail region. So many animals have sensory equipment or sensory tissue like nervous tissue that will have brain, nerve cords, spinal cords. And that's something we'll briefly look at the very end of the semester in the nervous system chapter. In terms of movement, radial animals are usually sessile or platonic, which means they're not going to swim very well if they swim. So usually we'll see that they're actually stationary, or at least semi-stationary. And then bilateral animals will usually be the ones moving about. So, in terms of tissues, in this chapter we'll look at some embryonic tissues, which will be next. But there are a few groups that lack true tissues, sponges being the most notable one. But in terms of development, there are three tissue layers that will, or germ layers is what we call them, that give rise to the tissues and different organs of that animal embryo. And so that's shown here. And this is something you'll understand. And so looking at these three layers, again, if you break down the word, you should be able to understand where it is, and then from there you can understand what it does. So ectoderm is going to form, it's going to be the outermost layer. So ecto meaning outer, derm meaning skin or layer, and so that's the outer layer, which is here. It will give rise to the epidermis and the nervous system for humans. For other organisms, it can form the exoskeleton, which is noted in the picture here. So this is going to form the outer layer. The mesoderm is going to be the middle layer. So you think even M starts with, they both start with M. And so that will give rise to pretty much all the organs, muscles, bones, the dermis of the skin, which is below the epidermis for humans. And so that is here. And then thirdly, the endoderm, the innermost layer, that's going to give rise to the digestive tract and other hollow organs like the lungs. And so that is going to be, all these are going to slightly vary based on the animal you look at, but you do want to know which these three are, where they are, and kind of what they give rise to for humans, which is noted here. Right, in terms of these germ layers, some animals will have all three, and those are going to be called triploblastic. These animals have all three layers. Some of the animals only have two layers. They'll only have the ectoderm and endoderm, and that will be called diploblastic. So I think like the prefix means two, like diploid, two sets. So this is two. And then triploid is, or triploblastic, that's three. So 2 layers versus 3 layers. We're moving right along into body cavities. As I mentioned earlier, that blastocele, seal, the suffix, and it looks like coel, but is pronounced seal, comes from the word coelom, which is here. I believe I'm not sure what it's called, but how you pronounce it in the book, it looks like that, coelom, if that helps you. This is a word that means true body cavity. And a true body cavity or opening region is going to be derived from the mesoderm tissue, or mesoderm germ layer. And so we'll see in the next image some different body cavities, and so the ones that possess a true coelom are called coelomates. And so that's something here. Yeah, and so this is one of several you'll want to understand. Just checking my slides here. All right, looking at coelomates, our example in this picture is an earthworm. And so coelomates can have a true body cavity from the mesoderm germ layer, which is shown here in red. And so if you look very closely, you can see that there's like an opening, kind of actually 2 openings, one on the left side, one on the right side. and it has a note mark with the word coelom where the cavity is. It's formed from the mesoderm tissue, which is in red. And so that's a true coelomate. It's going to have the mesoderm tissue forming a body cavity called the coelom. Now looking at the second one here, pseudo-coelomates, if you remember, I think that's actually not spelled correctly. Nope. I think that should be as pseudo-coelomate. And so that is a false coelom, or false body cavity. So that's what you see here in this, I believe it should be a nematode or a roundworm. They have a false body cavity of that's not completely surrounded by mesoderm germ layer. And so you can see that opening, that cavity within the animal. One side of it has a mesoderm layer. The other side is a digestive tract that's from the endoderm. So that's a false layer. So anytime you see pseudo, you know it means false. And so this is a false coelomate. Now, a coelomate, so last one here, you know the prefix, hopefully by now. A means without when it's in front of something. And so this is without a coelom. So there's no cavity here. And examples are nice, lovely flatworm we'll see in the next chapter. And so if you see that flatworm body, they have the ectoderm, the mesoderm, and then endoderm. And so the mesoderm doesn't have a body cavity or opening, and so that makes it a coelomate. So we'll see these words in the next chapters, and just want you to understand them now. That way when you see them, it's something you're familiar with. Right, and so here's also a good slide to know if you like words more than pictures. Pseudo-coelomate, acoelomate are here. And so it comes up to one of our last things here in terms of animal development. And the embryo, as it from the zygote grows into the embryo, we have two major differences in different types of animals. And so we've classified them by name into protostome and deuterostome development. We'll see these words pop up a lot, and so that's why we'll want to understand them. But it's also in terms of just general animal development knowledge. This is one of those key things that helped us organize animals based on their similarities and based on their differences as well. And so it's very important in terms of animals and animal classification. And so the difference comes through that cleavage, that rapid cell division that the zygote undergoes. There's a difference there. And for protostome animals, their rapid cell cleavage is going to be what we call spiral and determinate. And so looking at the cell as it divides and becomes a new cell is actually going to divide in a way that's going to produce a kind of spiralized form in the ball. And so looking at that, most notably here, is shown that the cell on top of a bigger cell, the end of the cell doesn't line up, and so it kind of, as it divides, it kind of spirals into like a double helical shape. And so that's what you see here is kind of a spiral going. And so that's kind of the physical difference, along with the capability of the cell. It will be the second part there, determinate. This means that the cell can only divide into or develop into certain parts of the embryo. And so comparing that to our deuterostome development, deuterostomes are going to have indeterminate cells, which means one single cell can make the whole embryo. And so that allows for identical twins to be formed, along with other things. But that's a huge difference between the protostomes and deuterostomes. Another difference here in the 8-cell stage for deuterostomes is that they have a radial cleavage, which means their cells are that the ends of their cells line up radially, which is hard to point out here. You can see they all kind of line up radially on the outside there. And so that was on this slide here. And so here, just noting the indeterminate cleavage, each cell in the early stages of cleavage retains the capacity to develop into a complete embryo. And so that allows for identical twins. Embryonic stem cells can forever grow and divide through this in this way. Under lab conditions, they can keep embryonic stem cells basically forever. So looking here, our examples, if you like to kind of classify protostomes, are mollusks and annelids, so shelled marine animals and worms essentially. Deuterostomes examples are echinoderms and chordates, which will be chordates have a what we call notochord, and that's something we'll look into. Basically branches into vertebrates and invertebrates. So most animals will probably be deuterostomes. All right, and also on here you can see some other parts B and C. These are also hugely important for development. And so looking at the next one is the coelom formation. So that is your body cavity formation. And so looking at our slide here, you can also use this. And that protostomes, their coelom forms from when the mass of the mesoderm split, as compared to deuterostomes, that mesoderm is going to bud off from the inner wall, the inner layer called the archenteron, and that will form the coelom. And so you can see that in the picture here, that the coelom mass, or the mesoderm mass in the protostomes will be split to form the coelom, whereas in deuterostomes, the coelom here is pinching off of the archenteron, which is that inner endoderm layer. But that mesoderm is that red part there that forms the coelom. And then the last structure that's important is the fate of that opening. So when the blastula pinches inward and makes the opening of the for the gastrula, that's called the blastopore. It makes sense, right? The opening of the blastula, right? The pore of the blastula. This forms during gastrulation and connects the inner layer of the archenteron to the gastrula layer. And so in protostomes, this opening will develop into the mouth, and for deuterostomes, this opening develops into the anus, which is just a difference, and so that is something that we will also want to know. Alrighty. And so the rest of this chapter kind of helps look at the phylogenetic tree and looking at molecular morphological data. It's just a few slides. I like to kind of go through it to prep you for the next chapters, but this stuff we will see in next chapters, so it's not necessarily something that's listed to review on this chapter. And so looking here, there are 5 important points about relationships among living animals, that they all share a common ancestor. Sponges will always be the basal animals, the basal taxon, or the outgroup of all animals, as they have the least shared characteristics with the animals in general. And then the animals that have tree tissues are called eumetazoans. Most animals belong to the clade Bilateria that form the bilaterian animals, which you'll see are invertebrates and vertebrates. And so looking here, this is a current phylogenetic tree. And in the next chapter, we will make our way down this tree from looking at these large groups of animals here. And so you'll see the words Bilateria, Deuterostomia, Lophotropozoa, Ectodisosa pretty frequently. But in terms of looking at these, I like to stick to like the general groups that we'll go through as we go through them down that way. And so here, the bilaterian animals are into 3 large groups: Deuterostomians, Ectodisosans, and Lophotropozoans. Deuterostomia forms the echinoderm, so sea stars. Also, hemichordates are acorn worms, and then the chordate groups. This will include the vertebrates and invertebrates. The Ectodisosans are a unique clade of invertebrates that shed their exoskeletons through ecdysis, and so that's where it gets its name. And then the Lophotropozoans are bilaterian invertebrates, and so they have a unique structure that links them together called the lophophore. And so they also have this lophophore stage can be in adult or the larva stage, and so it kind of depends on the organism. And so that's shown here, that the lophophore feeding structure on this ectoprot, something we'll look at next chapter, is a tentacle-like capturing food particle feeding structure. And then the larva is, as you can see, morphologically different than the adult and has some nice, lovely cilia projections around different regions there. And so in terms of animal systematics, there's always in-search, inquiry going on. And so these are some questions that are focus of current research. If you're interested in this type of topic, those are some good places to start with. And this last one is just a figure at the end of the chapter. So in terms of eras, while I don't focus on it, it's here for you. So, but I think that wraps up chapter 32. Please let me know if you have questions. But until next time, bye-bye for now.